Alright, welcome back in this video. We're over DNA bonding and complementary base pairing. I made a lifetime total of $45.12. You may notice the hype button has appeared on my channel. What this is if you happen to watch one of my videos that has been published within the past 7 days, you will see this button. It's only on the YouTube app, not on the website. Don't know why they did it, but that's just how it is. And what it is, it's a completely free way to support me. If we were to click this button, it gives my video points. And the more points that I have, the more popular the video will be. It'll be basically shown to more people and recommended to more people, which will help me out. So go ahead and please click that. That'll really help me out. In addition, please like and subscribe and leave a comment. All right. In the next 2 minutes, I'm going to blow your mind. I'm actually so certain of it, I would put money on it, even though I'm broke. I'm only at $45.12. But, I would put money on it. And I think you're going to learn something new. Okay, nucleotides, they're spelled wrong. I need to fix that. Nucleotides need to be able to bind to each other on the same strand and on the opposite strand. This is how DNA synthesis works. We're forming DNA. Nucleotides on the same strand bond together via phosphodiester bonds. Why is this important? Here's the kicker. When you learn something new, just pretend you learn something new. It could be something from this video, hopefully. You learn something new. Neurons form synapses with other neurons. Now, when neurons form synapses with other neurons, it requires a lot of proteins, many different types of proteins. There are scaffolding proteins. There's Sonic the Hedgehog protein. It's a real protein, by the way. Actually, in fact, the creator saw a magazine of Sonic the Hedgehog, like the game, while doing his studies or whatever, and that's how he named it. So it's a real protein. Sonic Hedgehog protein is a real protein. And a bunch of other proteins are involved in synapse formation. Okay, well, right, we said that proteins are required, but how do we make proteins? Proteins come from RNA. And you might be thinking, well, where does RNA come from? It comes from DNA. And DNA has phosphodiester proteins. It's everywhere. All the proteins in your body, DNA, even if you look, it's even in the outside world, for example. If you do a DNA testing, like the 23andMe, phosphodiester bonds are present. That's how they do the testing. If you look at someone's hair, there's phosphodiester bonds there. They're everywhere. That's why it's important. Nucleotides on the opposite strands must bond together in correct pairs via hydrogen bonds. This is called complementary base pairing. This is what you're usually you're probably familiar with. The A's match with the T's and the G's match with the C's. That's complementary base pairing through hydrogen bonds. And we're going to go over everything. Phosphodiester bonds are covalent bonds, and they're basically permanent. There's nothing that can really break them. There's only a few enzymes that can break it, but it's very hard. Hydrogen bonds are not that strong, and they're very easily broken. And actually, I'm going to give you a really good example later in this video. Okay, let's talk about same strand bonding. Same strand bonding is basically the nucleotides on one strand, like these two right here, how they stack on top of each other, basically. That's what same strand bonding is. Here's the trick. Super easy. You're going to have 2 of them, or 3 or 4. And what we're going to do is on one of them, so let's just take 2, for example, 2 nucleotides, 2 different nucleotides, or it could be 2 of the same nucleotides. It does not matter. The bases do not, it could be 5 A's in a row. It could be 3 G's and 2 C's. It does not matter. This does not matter because we're not doing complementary base pairing like this. This is just same strand bonding. It could be in any order. It does not matter. So this does not matter whatsoever. What we care about is the pentose sugar and the phosphate. On one of the nucleotides, erase the OH. On the second nucleotide, leave the OH, but move this bond there. Move the double bond over to the left. And that's all you need to do. When you do that, make sure you stack the one that does not have the OH on top, and the one that's you move the phosphate to the bottom and line them up together. So the 3 carbon, the third carbon is lining up with the phosphate from the second nucleotide, just like this. See how I kind of lined it up somewhat? This right here is the phosphodiester bond you just created. So what we did is we sacrificed an OH, took that out, and we moved the double bond and we connected the 3 carbon, the 3 prime of the pentose sugar to the 5 prime phosphate. That's what we did. So let me just write it here. Let me actually just do the actual reaction right now, just to show you how easy it is. Whatever, same color, so close enough. Okay, it was like this, right? And then, yeah. Okay, it was like this, roughly. Right, you have 2 nucleotides on top of each other. It does not matter which one they are. On the top one, erase this. Actually, you know what? Just erase the H. And then this one, I'll just erase this. And like that, leave it like that. And then bond them together. And that's how you do it. That's all you have to do for same strand bonding. Now the name, phosphodiester linkage. What does it mean? Well, it's actually in the name, right? We have a phosphate group. That's the phospho. And diester, 2 esters, di, meaning 2. We are forming an ester here and an ester here. That's why it's called phosphodiester. And it's a linkage because we're linking them. And that's same strand bonding. Not too bad, right? Complementary base pairing. Okay, so this is where it matters. This is where the A's must bond with the T's and the C's match with the G's. When you do A and T bonding, so on an exam, your professor may ask you, show me the bonding between adenine and thymine, for example. This is what you have to draw. Exactly this. And you get the problem 100% right. So you need to remember this, and you need to remember this. And all you need to do is make sure you expand the NH2. This is the NH2, right? So make sure you expand it so it shows the hydrogens. And make sure you expand this as well. And just literally bond these together. Like this. And that's your answer. That's it. I don't know what else to say. But here's what's really what's important here is the helicase. So whenever you do DNA replication, right, you probably learned that you have to rip apart the strands, right? The strands unwind basically and come apart. This is what helicase is targeting. Helicase shreds this apart. It separates them so they're in a single strand. So helicase is destroying hydrogen bonds. That's what's happening. And I'm putting this in a box because I actually got an exam question, exactly this question. The question was, between adenine and thymine, how many hydrogen bonds are there? And the answer is 2. You don't know how many people missed that on the test. People said 3, some people said 4, some people said 1. It's 3 points, as long as you remember this. All right, here's cytosine guanine. So there are 3 hydrogen bonds here. So once again, make sure you expand everything. If it has hydrogens, it's probably doing something. So whenever you see a hydrogen part of your functional group, it must be matching up with either oxygen or nitrogen. It's not matching up with another hydrogen. It's always matching up with an oxygen or a nitrogen. That's the trick. So make sure you expand it. If it has hydrogens, then it must be matching up with an oxygen or a nitrogen on the other atom, on the other molecule. And yeah, that's it. Just copy and paste this. Remember this. Remember what cytosine looks like. Remember what guanine looks like. And write this a million times. You'll get the problem right. I promise you. Until next time. Later.